All right. Ask them if they can hear me now. Are they able to hear me online at the moment? She can. Can he? Yeah. Yep. Okay. And I will. All right.
Okay, hi everybody. Thank you for joining us this afternoon. Appreciate you taking time out of your busy afternoons. My name is Lenny Bain. I'm the genetic analysis rep for ABI Thermo Fisher Scientific. So I help in solutions like qPCR, Sanger, digital PCR. We're talking about digital today. Um, Before we do get started, though, I wanted to thank the University of Wisconsin Biotechnology Center and the BioGarage for sponsoring this or helping us do this, hosting us. So they're a great group up at the BioGarage. They have several qPCR instruments that you can essentially rent. You walk in there, it's very affordable. So you can go in and do, if you don't have your own qPCR instrument, use their instruments. They also have a digital PCR instrument, an absolute Q, that Holly's going to be talking about here in a little bit. So, like I said, it's a great service. So I think that's all the general announcements I wanted to make. So I'll introduce Holly to you to get us started. Holly is our digital PCR technical specialist. She has a chemistry and biochemistry degree from Hope College, with graduate work in molecular biology from Indiana University. She was a scientist with 4 years of bench experience as a researcher in both academia at the University of Michigan and also industry with Pfizer. She joined Invitrogen / Applied Biosystems / Thermo Fisher over 24 years ago and has spent the last 18 years as a genetic analysis specialist, supporting qPCR sequencing and now digital PCR. And then in the last couple of years, she's moved over to this role as our digital PCR specialist. So, Holly.
Wow, that was a big speech for just I'm old. All right. So, thank you very much for joining us. Before we get started, I usually like to get a quick feel for who's in the audience. How many of you in the audience have experience with qPCR? Okay. How many people have utilized digital PCR? Okay. All right. So today, what I am going to do is I'm going to kind of go over just some of the fundamentals on how digital PCR works. And then I'm going to go into some of the common applications for the people that I work with and what kind of stuff that they use digital PCR for. So if you have questions, because we're recording the session, I'm actually going to go all the way through. There will be time for questions at the end. So if you think of something, write it down or remember it at the end for me, okay? All right. So we're going to get started. So today's seminar is just going to kind of go over the details about digital PCR. We're going to explain how it actually differs from qPCR, and then I'm going to go into some common applications. And at the end, I'm going to briefly introduce the Absolute Q, which is the instrument that is available here at the BioGarage for your usage. Okay, what is digital PCR? Well, it's really the division of a bulk reaction, right? So it's the division of a bulk reaction of nucleic acid targets. So I'm going to just have a moment. Sorry. Oh, I'm so sorry. It's the method of quantifying your nucleic acid by individualizing a bulk reaction. And we're going to do that by just dividing it into thousands of independent reactions. Now, most of you are probably familiar with PCR, in which we get to throw together master mix and assay in our sample, and we go through a bunch of amplification cycles to throw it on a gel and find out that our product is there, or it's not there, or worse, there's multiple bands and we don't know why. So that actually came out about 40 years ago. Real-time PCR was developed actually just over 30 years ago. What was neat about real-time PCR is they figured out how to throw in a fluorescent molecule so that they could actually watch the amplification cycling live. This enabled them to literally plot out the changes in amplification over time, giving you what we know as a real-time curve. The challenge, however, is that real-time PCR is a bulk solution. And so in that bulk solution of amplification, in order to make sense of everything that was going on in there, we had to be able to relate it to something else, hence relative quantitation. We relate it to an endogenous control, as you can see between the red and the green, or we'll relate it to a standard curve, which is in the gray. Just over 10 years ago, digital PCR was a technology that became readily available to the scientific community. It actually enables us to take a bulk solution of real-time PCR, spread it out over thousands of individual microreaction wells, and then interrogate on an individual level. So instead of a bulk solution, we're actually interrogating individual, which allows us to count the molecules that are present, giving us the absolute quantitation. So what is digital PCR good for? It's great if you don't have a standard curve for a reference or you want to get away from doing that. It's good if you're looking for a rare target against a sea of wild type or a background of wild type that overwhelms your qPCR reaction. It's especially helpful if you're looking for very small fold changes, and we will get into that at a later point. It's good for single molecule resolution or multiplexing if you're looking for multiple targets. But the thing I like it most for is it's really good if you're working with challenging samples that sometimes don't work with qPCR. So how does it work in terms of comparing to qPCR? Well, as I mentioned earlier, quantitative PCR is actually relative PCR. And so we have done a variety of things in order to make sense of that data. We've created a reference gene so that we could identify, well, how much sample is going in. And if my reference gene doesn't change, then I can actually calculate the fold change. But you have to make sure your reference gene is a good one. Otherwise, people make sense by choosing to do standard curves. The challenge with quantitative PCR or relative PCR is that we are very reliant on the efficiency of the reaction. We would love to base everything on 100% efficiency, but sometimes the sample won't allow it. Sometimes our primers are not 100% efficient. When that happens, it changes the slope of the curve. And so we are at the mercy of finding ways to justify that data. By taking that same reaction now and spreading it out across individual wells, and so each well is either going to have a molecule of interest or it will not have a molecule of interest. When we do that, it individualizes everything so we can literally pick out our positives instead of worrying about how they are all playing nice together in the sandbox. They can have their own sandboxes now. It gives us better precision, and we're no longer doing a relative curve, but now we're just doing an endpoint analysis of yes or no. So qPCR has a lot of value in research. First of all, it has a very broad range of detection. You can throw high expressing targets at it versus low expressing targets. You can do high throughput analysis. You can screen tons of samples. It has a lot of benefit in that regards, but as you can see by the graph up here, it starts to lose its precision as it gets to the lower end of your expression levels, right? When you get to the very low end and you have just a few copies, it's not very precise and it's difficult to get reliable information from qPCR. This is where digital PCR really comes into play. As you can see from the arrow, you're getting really great precision of the quantitation of your sample. However, you'll notice that the arrow got shortened. So as I mentioned, real-time PCR is 8 orders of magnitude, but digital PCR is going to have a shorter range. So when digital PCR was first introduced, the goal was about 20,000 partitions, right? 20,000 sub-microreactions. And so 20,000 gives you a range of about 5 orders of magnitude. That became known as the sweet spot of digital PCR. And so as you can see from here, if we have high expressing targets, sometimes beta-actin, GAPDH, they're not great targets to use by digital PCR. But everything else that falls at about 25 and lower, just what you need if you need greater precision, especially if you're working with anything over 30. A CT of 30, it's a great way to flip over to use digital PCR to get what you're looking for in terms of precision. All right, so, I didn't mention it, but I will talk about it. Monitoring small fold changes over time. So qPCR is known for being able to be sensitive to a 2-fold. And if you're using a Quant Studio, you can probably get down to 1.5-fold change of significance. If you're looking for something even smaller than that, right? Because we've all been doing research a long time. So then that means we've covered the big targets. Now it's time to dig into the smaller targets and find out what's going on. And so this has the ability to fine tune that for you. This is another representation of how digital PCR has, or I'm sorry, real-time PCR has a broad range, but the precision's not that high. Digital PCR, while smaller range, it is much more precise, but you'll notice that it has the ability to get into a range where even qPCR can't reach. So this relates to the fold change question. qPCR and digital PCR should essentially give you the same answer, right? We're just quantifying the molecules. The challenge is with qPCR, we run duplicates or triplicates in order to get the average, right? Because it's a bulk solution. There's a lot going on there. So we run replicates to make sure that we're in the right ballpark. So while we're running replicates, we're actually increasing our error margin. When we move to digital PCR, because we are literally counting the molecules that are present, we actually get a much more precise answer every time. And so our error margins tend to shrink in digital PCR than they would with real-time PCR. This is why if you're looking for small fold changes, it's better to use digital PCR because they would fall within the error of margin in qPCR. As you get to lower expressing targets, it becomes all the more impactful when you're working with low expressing targets. All right, so this is just a concept of how it is that we actually collect the data. So I talked earlier about the efficiency of real-time PCR. So if I have two samples, same target gene, mind you, but if I have two samples and one of them is efficient, say 100%, but one of them is a little less efficient, so you end up with more of a sloping curve between the two. Well, the threshold is arbitrary. We decide where to put that. Well, maybe the machine decides for you, but we decide where to put that threshold. Here's the problem, though. If the threshold is here, that's one delta CT, but if the threshold was moved higher, that's a different delta CT, or downward, etc. You get my drift. It's where we put that threshold that impacts the way we interpret the data in qPCR. In digital PCR, we're no longer interested in the efficiency of the curve, the bulk solution amplification. We just need to know whether it amplified or didn't amplify. So now we're actually taking our reading at the endpoint as opposed to here. This means if there are any amplification inefficiencies, digital PCR doesn't care anymore. We're still able to count your molecule. So here's an example of a qPCR plate. Of course, if we have sample in our well, it's going to amplify over time. We're going to plot it out, and either we have product or we don't have product. In digital PCR, we're going to take the same well, but this time we're literally going to spread it out over thousands of individual wells. And now our target is either our molecules are either going to land in a well or they're not going to land in a well. So now when we go through amplification, we actually either see amplification or we don't see amplification. And so you can see anything with a green would be where our target landed, and the ones that are black are where we have nothing. If we have more dots, it's a higher concentration. Fewer spots, lower concentration. But how does this actually translate to our copies per microliter? So this is an image of the array from the absolute Q. As the sample is put across here and amplified, it's going to come up with a response that shows, yes, you have the target that you're looking for. This is an inset in which we can kind of see those. We literally count the dots and the fluorescence, and then they are plotted on a chart. So just so that you understand, if I have 20,000 wells over here, each dot represents a single well. So in terms of this one, this is a high fluorescence, it would be plotted up here. This one over here is a low to no fluorescence, it would be plotted down here. So as this goes through, each dot is a single well. Now it enables me to actually identify how many are the positives I'm looking for, and I can count them to get my molecule count. However, the number of positives does not always equate exactly to my concentration of copies per microliter. The reason for that is because I have to take into account Poisson statistics. I can't guarantee that every well has exactly one molecule. I can guarantee the none, but I can't guarantee the one. So how does that work? In case you're not familiar with the way Poisson distribution statistics operate, that math has already been determined. So if I have a grid with 100 squares and I have a bucket of 100 balls and I dump the balls across the grid, I would love it if I had one ball for every grid. We all know that's not the way the ball bounces, right? So what happens then is the majority of them may have one, some will have none. Occasionally there will be twos and threes. The challenge, of course, is that as my molecules go across the well and they flow so that I have ones and zeros, twos and threes, once I go through amplification, all I have are zeros and ones, positives and negatives. So I look at this and I can count, but it doesn't actually perfectly reflect. Well, the good news is the Poisson distribution statistics have already been established. And this is the way that every digital PCR machine operates in the math for calculation. So theoretically, I have a 1-to-1 relationship, or a 2-to-1, 3-to-1, and in the reverse, a 1/2-to-1 relationship. Now, I can't tell you exactly how many I have here, but I will say this. I can always tell exactly how many zeros I'm supposed to have in order to have that distribution. And so what that means is that as we do digital PCR, it's very important that we put the right amount of sample on there so that there are some negatives that we can actually apply Poisson distribution math to and come up with your calculation for copies per microliter. So what are people using it for? Absolute quantitation, copy number of gene expression, molecular integrity, which is an interesting new concept. SNP genotyping, we will talk about that. It's an interesting shift from qPCR. Rare target detection, very popular to find that proverbial needle in a haystack. But people use it often in cancer research, cell and gene therapy, a lot of environmental testing. And then genome editing is a very common application. All right, so absolute quantitation. This will give you a lot of insight on how digital PCR, the specificity of digital PCR works. So people who use standard curves for absolute quantitation in qPCR often have to find the standard. So they have to find one and have to make sure it relates to the particular target that they're looking for. Sometimes you're looking at a synthesized standard versus a naturally occurring organism. So you get stuck trying to figure out, is it relatable enough? Does it respond the same way? Or can you even find it, or do you have to make it yourself? So the challenge then is that hoping that it's accurate, especially if you bought it from a company. Let's hope that they're using digital PCR to quantify it. And the repeatability. But I think the biggest challenge with doing a standard curve in qPCR is that it takes up a lot of space on your plate. And so you've devoted almost an entire row, if not more, just to the standard when you could be running samples instead. So this is actually what a dilution series looks like on a digital PCR. What I love about this is I mentioned earlier that you can actually get 5 orders of magnitude. When we're dealing with 20,000 partitions, you can get up to 5 orders of magnitude of sensitivity. But what's really nifty here is you see we actually did a dilution curve for you just to demonstrate the precision of digital PCR. Now over here, this is actually, it says 10,000x. It's actually probably close to 76,000 molecules. So as this is put across the array, the majority of the wells are actually positive. There's a few negatives, just a few, but this is what we would define as the very high end limit of digital PCR detection. Now, in this particular case, the majority are filled, but because there are a few negatives, I can still give you quantitation. As I go through the 10-fold dilution, you can see there's a shift from the positive cloud down to the negative, and that continues on the way down until you get to the other side of the coin where you have just a few positives and mostly negatives. Now, looking up at the standard curve that's drawn on the top, what's fascinating is through the middle section, the precision is spot on. Even at the highest end and the lowest end, where the precision varies just a little bit, right, because we're at the very limits of the technology, the precision is actually still a whole lot better than it would be at qPCR. And so if you're looking for absolute quantitation, digital PCR is the way to go. So let me tell you about some creative ways that people have used digital PCR. Maybe it'll get you thinking a little bit about your project. So we had a lab come to us, and they were actually doing some viral work, and they were trying to figure out whether or not their viral genome was intact or not. They were trying to figure out whether it was virulent by the time it was introduced to a new system. So they had to develop a way to determine whether or not it was being degraded. And so we worked with them to design actually a series of assays of different colors all the way across their stretch of the viral genome. So the concept was if the virus was intact, all four colors would actually be together on the same molecule. They would show up in the same well. Whereas if they were degraded, then you would get some or none colors. The concept is it would look like this. So as the sample is washed across the array, spread across the array, I should say, as the sample flows across the array, your molecule will land in a single well. So if it's intact, all four colors versus degraded, some or none. Bigger representation of it. This is actually a fun little output of the results. This is not just typical results, but this is actually the color of the fluorescence in each single well in the array. What I want to bring your attention to here is that there were 166 wells that had only FAM, only Vic, only Abby, but when you found the ones that had all four, this particular sample actually happened to be a degraded sample, so it had very few that were all four. But it's a nice little trick of the software that allows you to actually do projects. In fact, I do work with several labs that are very interested in finding out how many wells have both of their targets in them together. So it's a nifty trick of the absolute Q. Copy number. So copy number can easily be done by qPCR, but if you're looking for a non-integer or a more sensitive method, especially if you have higher copy number, digital PCR can be much more helpful in that regards. This is an unusual example, again, of a customer that came to us, and they were doing some genome editing project, but they were doing an insertion of a gene, and they wanted to see where it was being inserted. Now, they had run it on qPCR, and they came up with a target of 3, but they also ran it on digital PCR, and as they found out, there was only 1 copy. And they were very confused as to what was going on until they realized that maybe we can check this in a different way. They ran the sample through a digestion, and what happened was it changed the result to 3. And so they found out that in their process of editing, they were actually getting an insertion in tandem across the same molecule. And so it's a unique way of using the absolute Q digital PCR just to kind of understand what's going on. Now, this isn't even the genome editing that we will talk about, which will be later. So I mentioned earlier that one of my favorite things I like to use digital PCR for is overcoming inhibitors. So as I mentioned earlier, in qPCR, we're extremely reliant on the efficiency of a reaction. So if I have 2 samples that actually have the same starting amount of material, but one of them happens to have inhibitors, which slows down the amplification, versus one that doesn't, it's going to give me a different CT result because I'm very reliant on where that sample crosses the threshold. However, since we started with the same amount and I'm getting different results, I'm making the wrong assumption when I look at the data. Digital PCR is now looking only at the endpoint result. So while these are all individualized curves, even if the curve is slow to get started, as long as it crosses the threshold, now I get to count it and identify it as being present. That means the digital PCR is much less influenced by inhibitors. Yes, it could slow the amplification down, but if it crosses the threshold, I get to call it as a positive, and my accuracy is increased significantly over what my qPCR results would be. These are some of the labs that I work with. Actually, because you guys are Wisconsin and you border a Great Lake, this is actually E. coli samples from Lake Michigan. Actually, it's a river inlet from the Michigan side. We test for a lot of E. coli, and I actually know that the labs over here also test for E. coli regularly on the beach areas. And so what I thought was interesting about this particular sample is at the higher concentrations, there is what we call rain. Environmental samples have a lot of inhibitors in them, and it's difficult to extract them. So I mentioned that it impacts the efficiency of the reaction. Well, now remember, this is on a scale of, say, 0 to 100 in terms of fluorescence. Well, this positive here wasn't quite 100% amplification efficient, but it still crosses the threshold, so we're going to count that as a positive. What's intriguing is the same sample over here is just a 10-fold dilution. You'll notice that as we go through the dilution, you actually get rid of a lot of that, which means we've also diluted the inhibitors that are present. But as I come over here, you can see by the dilution that the actual precision is much tighter here, and we're getting such tight replicates. because we have such clean results here. So the point of it is that even from a 1 to 10, your precision increases, but you also eliminate some of the problems with the inhibitors. All right, this actually is a sample from a fish hatchery, and they were looking for the invasive mud snails. And so he had a qPCR protocol in which he ran out to 45 cycles. I personally wouldn't believe anything over 35, but his cutoff was actually 38. And he came to me and he said, I'm getting a tail that's coming up at 39. And I think there's something in there. And I was like, okay, so we can look at that. But what I would like to bring your attention is he was right. It didn't cross the threshold. He couldn't call it a positive, but when we ran that sample on digital PCR, we clearly saw the DNA that he was trying to find in his water sample. He could not get it to come up in qPCR because there were so many other inhibitors in the water. When we ran it against the control, which came up perfectly clean, it gave us the confidence to know that indeed his sample had invasive mud smell in it. So now he uses digital PCR for the samples that don't pop up automatically. He double checks them with digital. SNP analysis. This is another one I think is very fascinating, especially, does anyone in the room do SNP analysis? Okay, you might find this very interesting then. So a SNP assay is designed in a way that it's actually, it's a single assay, but it has two different probes. It's looking for what would be deemed the wild type versus the mutation. So basically, these two probes are identical to each other. They're labeled with different colors. So the theory is that you would get a response from one or the other or both. So of course, your data usually looks like this. So SNP analysis is actually endpoint read in qPCR. The reason for that is what we call the clamshell. So if you're doing the real-time aspect of it, the clamshell means that if you happen to be a heterogeneous sample, you can't quantify how much of one or the other is in there. You just know that they have both, because the curve could actually swing this way, or the curve could swing this way, which means it has more of this one or more of this one. All you know is it has both. So quantitatively, we can't actually give you the data you're looking for. However, digital PCR has a unique way of resolving that data and giving you what you're looking for. And so here, with SNP assays, as you're trying to dig out maybe the lower expressor of the two, and this slide's going to come back again in the future, it's in the wrong place, but the SNP assay, the whole concept of digital PCR is to individualize what we're looking for, so we can literally count the individual molecules. So I think the slide will come back in the future, but the point is, especially if you have a rare SNP, just by changing the ratios, instead of a 1 to 1,000, we're changing it a 1 to 1 so that it's easier to find. So this is actually what I was hoping to get to here, is SNP assays, no matter how well you design them, will occasionally have cross-reactivity. What you may not be able to see from there is this blue assay, and unfortunately, I hate showing COVID data, but this is a great example of 3 different variants. This probe at the top is for the Omicron, but the green one has a single nucleotide difference compared to the blue, and this orange one has a single nucleotide difference compared to the blue. Now, the green and the orange actually have 2 differences. So the likelihood of overlapping between those 2 is very low. What I find interesting about digital PCR is that, yes, the blue one actually has detection of both of the other sequences because there's only a single nucleotide polymorphism difference, right? However, it has less efficiency for the mismatch. So while it is detecting the mismatch, it is doing so with a much less efficient reaction, which means instead of getting a nice high positive band for the target that I'm looking for, I actually get a secondary band. This is very common in SNPs. Now, this is why in qPCR, it's just a qualitative approach because all I'm getting is a single signal from both of those. But in digital PCR, you are actually able to set the threshold above the less efficient target, giving you the ability to quantitate your target of interest and separate out the 2 signals. So I actually find that pretty cool. All right, gene expression. How many do gene expression? Okay, so gene expression, the rules apply across qPCR to digital PCR. if you are utilizing a reference gene in qPCR, you're probably using it for reasons of normalizing your DNA or your input DNA or some kind of frame of reference against different genes. When you switch to digital PCR, we still need to find a way to normalize your input DNA. And so if you don't have a better way of doing that, then we have to help you find an internal reference control. But the benefit of utilizing digital PCR for gene expression is helpful if you have very low expressors that you're looking for, or if you're looking for very small fold changes. So like I said, the concept is the same. The challenge, of course, is making sure that our reference is within range of our targets of interest. And so we can totally use things like GAPDH and beta-actin, but they have to be diluted at a higher level in order to fall into our sweet spot. The majority of the genes that we look for in digital PCR usually are lower expressors. And so each project is individual, and I am happy to discuss specific projects with you, especially if you would like to try one at the Bio Garage. We'll help you get started with that. But in the meantime, we need to be able to make sure that everybody is detectable. And so if you happen to be working with the common targets, human, mouse, or rat, we have this fabulous panel of 32 different endogenous control genes, and we can help you select one that is within range of the targets that you're looking for. Additionally, I found out just last week that there is a fabulous endogenous control gene tool in which you can put in your disease pathway or your species, and it will give you all kinds of suggestions that you can look at. So there are some great tools on our website that can support if you would like to change what you're working with. I had 3 people just last week come to me and say, I've been using beta-actin and GAPDH and averaging the 2 of them forever because they vary. It's like, we need to find you a new control. So now, coming back to this, we need to find genes that all play nice together. This is a fabulous trick of digital PCR. So for anyone who has tried to multiplex in qPCR, it's kind of like having a bunch of toddlers in a sandbox trying to play nice with each other with a set amount of toys, right? The strongest toddler is going to take the best toys, and it leaves the other stuff for the weaker ones to sit around and go, oh, yay. So what happens then is when you do qPCR and everybody's trying to play nice together, what you have is the strongest toddler amplifies as expected. The less strong targets will start to peter off, which means they cross the threshold later. In order to actually make qPCR work, there's a fair amount of optimization involved, and we do what's called primer limiting, which is basically tying one hand, wrong arm. It's basically tying one arm behind their back so that they can't use up all the reagents. In digital PCR, we're actually going to take all the toddlers and give them their own sandbox. And now I don't care about primer limiting. I don't have to have anyone play nice. I'm going to give them as many toys as they want. So no matter what color they are, they can choose the toy that suits them best, and they can amplify in the well so that I can pick them out. So that's why digital PCR actually requires very little optimization. We don't have to get everybody to play nice together. Not to mention, coming back to the same point that I made earlier, even if there is a delay in amplification for whatever reason, you've got a toddler that doesn't want to share in a well with another one, as long as it crosses the threshold, I'm still able to count as the positive. Remember, we're doing this on an individual basis. So the concept is basically like in qPCR, I have to relate it to something else. I don't have to do that anymore. I can look at as many things as I want. Well, 5. I can look at as many things as I want and actually get just an absolute quantitation for them. Job done. All right, this is an array, single array. Actually, it looks like 4, but it's literally a single array analyzed for 4 different targets at the same time. This is actually the data from each of the targets. And what I think is great about this is that the quantity is not compromised. Just by throwing it in the well, I can increase the concentrations, I can decrease the concentrations, and the quantity is not compromised in digital PCR. So, all right, genome editing. So there is actually a wonderful testimonial, and I think I'll probably see it. Actually, I'll wait till I get it on the slide. So genome editing is traditionally done genome confirmation for genome editing is traditionally done 2 ways. The first off is depending on the kind of genome editing you're doing. If you are doing a cut with an insertion deletion, you will invariably be using NGS because you don't know what the sequence is that you're looking for or what's happening. And so you're going to use NGS to find that. If you know the particular edit you're trying to do, then people generally use Sanger to go ahead and sequence what they're working with. The challenge is that Sanger has a relatively low limit of detection. Not to mention, it probably uses a fair amount of samples. So the difference here is digital PCR can do the same thing as Sanger, right? Because we're looking for a specific mutation, but the sensitivity is much higher, which can be very beneficial if you are working in a system that has low editing efficiency. So if you're trying to figure out how much is edited, or if you're really trying to just do some verification and you want to use a really little bit of sample, because once you do that editing event, you don't want to use very much of your sample before you move to the next step, right? So digital PCR can be a great tool to do that for you, and it can be a nice, quick, and easy answer. So this is an example of someone who was actually doing CRISPR-Cas9. They were doing a cut site with an insertion deletion. So while they didn't know exactly what the end result was going to be, they needed to figure out how they could actually identify whether or not their event was occurring. And so in gene editing, there are two places where you actually want to do your verification. It's when you first do your editing and you're trying to check the efficiency, but also after you do your clonal expansion, you need to do your verification. And so the project that was on 2 slides before this, they designed it so that they have their cut site, and then they have a control probe all within the same stretch of DNA. And so they were looking for genome efficiency. They were not as concerned with what the insertion deletion part of it was. They did use NGS for the other aspect of it, but this was the way that they verified if the editing event was happening. And so in the wild type, they ended up with both probes present, whereas in the cut one, they only ended up with the control probe and not the 2. So as they monitored their samples together, as they monitored their samples for genome efficiency, they were able to quantify how many were cut versus how many were still intact. So it's just the way that they did it, but it would be the same concept if you were looking for very specific mutation events compared to the wild type version. And what I was going to mention was that your very own Dustin, what's Dustin's last name? Thank you. Dustin Rubenstein actually spoke for us last year, but he has a testimonial on our website about how he used the absolute Q for genome editing efficiency project. So pretty cool. All right, so low limit of detection, very small amount of incoming DNA. All right, so just to give you some examples, we already talked through the DNA editing. We also support RNA editing. Is anyone doing RNA editing work? Okay, just to let you know, it can be done, and digital PCR is just as sensitive for that as well. This is the same kind of an example where they were looking for the editing events, and the sensitivity was there so that they could identify it. Okay, so this is the other big usage that people like to do, finding the proverbial needle in the haystack. All right, so rare target detection. It can be really, really hard. I often think of it as like a pile of yellow tulips with a few red. It would be difficult to see the red if they were just in that huge pile. But if I spread that pile up and then hang a picture on the wall, I can literally pick out my red tulips. So this is kind of how digital PCR works. It's great if you're working with single cell analysis or if you're working with residual, but it's very common in oncology applications, or if you're looking for a very rare SNP, maybe a disease state situation in an in vivo model. All right, so we are going to talk about liquid biopsy as a whole, but the concept again is it applies to just about any scenario where you are working with samples in which you expect really low mutation efficiency compared to the wild type background. And that is basically the same concept. So here, we're going to talk about how digital PCR, the reason that we can find that needle in a haystack is because of the partitioning. So we're back to the slide that I didn't realize was duplicated earlier. The concept, of course, is when you're looking for a really rare mutation, the relationship might be 1 to 1,000. It's super hard to find those two in there without having there be some degree of domination. You saw the data earlier for SNP on qPCR, right? It may detect these two, but you won't have any idea what what the quantitation is of either one. By changing the dynamics and actually partitioning the sample, you change the relationship so that it's a 1-to-1 or even a 1-to-0. So now we have the ability to literally see what's going on instead of just kind of hoping that we can get something there. The benefit, of course, is now we see the amplification individually. I can literally pick out the mutations. What's neat is digital PCR has been validated down to 0.1% detection mutation allele frequency. In addition, I have customers that use digital PCR to get down to 0.01%. It takes a little bit of effort, but that's just to tell you that the power of digital PCR can get you where you're going if you are working through really low frequency targets. All right, so I am going to kind of talk about liquid biopsy as a whole, because I don't know if you work with in vivo models and you might work with clinical samples, but taking a direct biopsy is very invasive. And so even if you were working in a tumor model and you understand, getting a biopsy, we use the biopsy to tell us what mutations are present. But how are we going to monitor the effectiveness of, say, treatments or other things that we do that change that genetic makeup? This is where we use what we define as a liquid biopsy, or a sample taken so that it is a general representation of shed tumor cells into the system, or shed mutation cells. But the liquid biopsy is non-invasive. We're taking a blood sample and we're going to be able to analyze. The challenge, of course, is this is a pure sample and has lots of what we're looking for. This one's a very dilute sample that has wild type background as well as just a few of what we're looking for. So our percentages have changed dramatically between the two. Well, you can't look at that by qPCR, but you can look at it from digital PCR. This is an interesting study that was actually published for juvenile myelomonocytic leukemia. Sorry, the I trip over every time. They published a paper in which they were monitoring some patients for cancer. And what happened was they went in, they took a biopsy, they did NGS, they picked out some very significant biomarkers from the panel, the cancer panel, and they defined those in digital PCR or qPCR assays, right? So they're using digital PCR for this. What they did was they took a blood sample after the first treatment and they checked those biomarkers, and they found that there was not only a decrease, there was an increase. So the second treatment actually had significant impact on the patient. So they're not doing additional biopsies, they're just literally monitoring the blood for the biomarkers. And so additional treatments actually had, I think this is the hormone therapy, had a significant impact, and it actually brought the patient into remission. And now they do blood tests just to monitor over time to watch that there isn't any recurrence. And so it's a great way to kind of find those needle in a haystacks and make sure that whatever treatment or experimental condition you're doing has impact. So this came up earlier, but the SNPs, of course, are designed so that you have two of the same probe with just one genetic difference. We have a lot of bioinformatics. We have a lot that are already made for SNPs, but if you are working on a project and doing something novel, we can certainly help you design something that will work with digital PCR and qPCR. The concepts are the same. These are just some really great data points from some of the assays we already have validated. And please, if you're not oncology, that's fine. But my point is that they've all been validated down to 0.1%. We have great luck with designing the assays so that you can get down to really low frequency levels. Additionally, what I find fascinating too is this was also a liquid biopsy, but because you're looking at very low frequency, this is a scale for qPCR quantity, and this is a scale for digital PCR. This tells you how much more sensitive digital PCR is. qPCR called it at 12,500, but digital PCR identified 22,000. So the concept is digital PCR is just that much more sensitive as a tool, especially if you're trying to properly quantitate. We have lots of assays available. If you're not oncology, I won't bore you, but if you are, feel free to talk to me afterwards. We have tons that are designed, and we can design more. Okay, so I've covered a lot of the common applications people use it for. If you're not already thinking of projects, please do so. We can talk about them afterwards. In the meantime, let me introduce to you the Absolute Q. This is currently upstairs on the second floor. Okay. So it's upstairs on the second floor. It is open access. And so if you'd like to use it for a project, I'll walk you through the parameters of your project and let you go. All right. So when we first designed the Absolute Q, we went out and talked to labs about digital PCR that was out there. We said, what do you like? What do you not like? They had a nice little list of things they didn't like. So they gave us a list of some challenges that they encountered working with other systems. And so the first thing that they didn't like was that it used too much of their sample. So you have to create really large volumes, but it's only analyzing about half of your sample. So you're losing half of your sample and you're using extra reagents that aren't being used. The other challenge was limited or inconsistent compartmentalization. We talk about the digital PCR sweet spot. In order to get 5 orders of magnitude of range, I like to have a big range so that my sample can fall anywhere in there. If it's a little higher, a little lower, I've got some wiggle room. The recognized sweet spot is about 20,000 partitions. The droplet-based system in particular shoots for 20. Sometimes they get 12. Sometimes 12,000. Sometimes they get 13,000. A regular experienced user sees about 18 to 19,000. If you have one sample that's at 18 and one's at 12, you're not getting the consistency to be able to compare the two. We might as well be doing qPCR and doing replicates, okay? So the challenge, of course, is they needed consistency. The next one is that the other systems require multiple instruments, and it takes a long time to process. But last and most important, and this is unique only to us, other systems are endpoint only. That means they do all of the thermal cycling. They partition, but then they thermal cycler, and then they move it and take a reading. So that means everything positive they see is positive. We'll talk about what that means. All right, so when the absolute Q was created, you have a bunch of applied biosystems engineers that have been doing qPCR for a long time. And so they're all about efficiency. First thing they did was, we want to use a little bit of sample, and we're going to analyze every drop, because if you have precious sample, we don't want you to waste it, but we also want to know everything that's in there. So we actually use over 95% of your sample. We also have such a simple array-based system that your sample flows in there. We see over 20,000 reactions every time. There are actually 20,480 reaction wells, but we consistently see over 20. If you see less than 20, there's a problem, and we're going to help you with that. The other part is it's super easy to use. It's just like using qPCR. You're going to make up your reaction, you're going to put it in the plate, you're going to walk away. It's also quick to results like qPCR, hour and a half, and you can multiplex up to 4 different targets in the well. The last but not least, and the one that I think is the most important, is Applied Biosystems has it all on a single block. So you are literally going to put your sample in, you're going to put it into the block, you're going to walk away. It literally pushes the sample into the array, does all of the thermal cycling, actually monitors during the cycling just to be on the safe side, and then it does the final read. But the benefit is that while it's going through the amplification, we actually take a couple reads during the run. We try to identify any wells that would be fluorescing before your amplification would get started, and we actually reject those wells from your data. Because other systems do their amplification off of the reader and then move it to the reader, anything that's fluorescent is a positive. That means that if there's dust, dirt, something else in there that's amplifying, or not amplifying, but actually reflecting fluorescence, that's counted as a positive. Our system won't do that. And so you actually get more accurate results. All right, the question is how does that compartmentalization work? How are we dividing your sample into individual reactions? Well, as I mentioned before, you're basically making qPCR reaction, master mix, sample, primers. We're going to spread it out, and we're going to amplify, and then, of course, take the reading. But how do we get it there? So this is a picture of our current plate, our original plate, which is the MAP 16. What I want to bring your attention to is a few things. It's a very simple platform. It is divided in columns of 4, which means that depending on how many samples you have to play with, you can run just 4. You come back another day and run another 4, or you can run 8 and then do another 8 tomorrow. The point is that we give you the flexibility to run as you need it. So other systems, if you want to run a plate, you got to run a whole plate. You can't do it again. You can't use it a second time. So if you don't have enough samples to put into it, you're going to have to wait until you batch up your samples to be able to run digital PCR. Here, we allow you to reflex in the sense that, oh, I have a couple samples, I want to see what's going on. I'm going to be able to run those, and then I can come back and use the rest of that plate at a later time. Additionally, I mentioned that we see consistency for 20,000 reactions every time. As I mentioned previously, especially in comparison to the droplet-based, you get a variability on the droplets, and so it's more inconsistent. Also, if we did a dilution, we still see a consistency of error margin, which is really spot on, whereas you see more division of the answers for that. All right, so this is what it actually looks like up close. So this is an image of the sample well. This is the bottom of the sample well. And as the sample goes into the well and we apply air pressure, it pushes the sample through the microchannels until it fills all of the individual wells. For those who have qPCR experience, you might be familiar with utilizing rocks as part of the master mix. In qPCR, we love to use rocks. We call it a normalizer, but I also think it's the great equalizer in the sense that if something happens to rocks, that's like a red flag that something's going on to your sample too, right? I use it as kind of a background, like to catch things that might happen. But we also use it in a way to like kind of normalize how much material is going in there, how much master mix, efficiency of reactions, etc. Here, we do not use rocks as a normalizer. We use it as a visualizer, which means that we add rocks to your master mix so that as the sample flows across the array, we can actually identify how many of the wells got your sample, right? We can tell by rocks and all the ones. At the very end of the amplification, where your positives actually light up, as you can see it by the green, we need to be able to compare how many positives versus negatives. Well, rocks helps us define how many wells are filled, and then we count your positives against that number. And so that's how we can apply our Poisson distribution. It's also a closed system so that any sample you put in here, there is no cross-contamination with anything else. And so you shouldn't see any kind of bleed over between the samples. Okay, this is a super quick video. It just shows you how easy the workflow is. Our particular researcher here, she had already prepared a qPCR reaction mix, and she's going to put it into the plate. They're going to show you another angle so that you can actually see how big the port is to put your sample into. You're going to load only 9 microliters, and then she's going to put an isolation buffer on top. For those of you that are old school, like me, it's the isolation buffer is like an oil layer. It prevents evaporation of your sample, but it also creates a density barrier. So it is a different density than your sample. Your sample will settle to the bottom, and the isolation buffer sits on top so that when the air pressure is applied, it pushes your sample in first. As I didn't see on the video here, once you are finished just loading your sample with the isolation buffer, you just press the gaskets right on the array, and super easy, not fussy at all. And then you put the sample into a plate right into the block. The plates are barcoded so that I talked about not having to use all of the plate at once. The instrument recognizes the barcode. So it knows which columns you already used. So if you forgot, you can put it into the well or you can put it into the plate and it will say, hey, you still have these two you can still use. So I think that's really a great feature. All right. We're almost at the end. Super easy workflow. So if you want to give it a try, let me know. We'll get you started with it. There is a slight change in the master mix. So qPCR master mix is a 2x. Digital PCR is a 5x. The reason being is in qPCR, half of it is master mix. So we tend to use larger volume reactions. Here, because digital PCR is often used to find the proverbial needle in a haystack, sometimes we need to load more sample. So we changed the master mix to a 2x so that it leaves more room for the sample if we need to load it. So that's about the only change. We use the same primers that you're already using. We would load it into the plate, run it, and get your results. Software is very amenable to utilizing whatever protocol you have that's working well for your primers. As you can see, we can do up to 4 different targets. There are some other selection choices depending on the dyes that you've got. We can start with RNA or we can start with DNA, whatever your preference is. And if you have some unusual primers that we need to play with, sorry about that. If you have some unusual primers that you need to play with, we can adapt that with a 2-step versus a 3-step. In addition, I mentioned the plate's barcoded, so you can define which wells you want to run, and you can come back and use the rest later. And what I think is really great, again, is we're going to come back to the fact that we're the only ones that do what we call a pre and a post PCR image. So we actually monitor the plate during the run so that we can extract anything that's actually a false positive. In addition, we do check for, oh, it's in another page here. In addition, we check to make sure that rocks looks good, is filling the wells properly. There's a lot of AI checks that we go through to make sure that all of your well data is really great. I talked earlier about getting better precision than 0.1% mutational allele frequency. To do that means that you have to analyze more volume of your sample. So like if you come back with one positive, you're going to run that across several arrays. If we come up with 4 positives across the arrays, then we can actually pool that all together and start to give you some significance for the mutational allele frequency. But this one allows you to pool. Additionally, the new software version that just came out allows you to study functionality. Anyone who is used to using a Quant Studio software, one of the things I like about it is if you're working on a project that takes place over time, you want to be able to analyze that data together. Most of us all just export to Excel and spend hours putting it together and figuring it out. The Quant Studio actually has study functionality software, so if you ever want to try that. Alternatively, here you can plug in up to 384 samples worth of plates, and you can analyze all of your data together, set common thresholds, look at project over time, all kinds of things. So I do appreciate that about software. And I already said that it does all of the checks and balances, right? ABI engineers, they're very analytical, so they want to make sure your data is as good as possible. All right, that's pretty much it. Super easy to use. It's flexible with how many samples you run through. Here you go. If you like podcasts, this can be fun. It's a bunch of goofballs, but a lot of them are our customers that actually use the Absolute Q for a variety of different things. So our website has that. It's available on Apple, Google, Amazon Music. Okay, thank you for your time. I really appreciate it. Do you have any questions? Are there any questions? Yes. Yes. So you may not be aware, but you have the right to set the threshold in the qPCR too. So the difference is when you look at digital PCR data, setting the threshold, the system will automatically set it for you. It actually looks at the negatives and it will set it between the first break between the negatives and the positives. Sometimes you get samples that are ugly. They look a little different on qPCR, but on digital PCR, if it's an ugly sample, you've got color everywhere and you've got rain. So it doesn't always do a perfect job for that. So you have the opportunity to move thresholds around. I personally like to have a singular threshold across all my genes, or I'll end up throwing that sample out. I like uniformity, and I like to be able to compare. Now, when it comes to the one SNP example that you're looking at, you would probably run your initial experiments to understand where that mismatch product was occurring, and you can set the threshold. What's really nice about the system is then you can set up the parameters that say, I'm using this gene, and I need the threshold set at this point so that it negates everything below and counts everything above. So you have the power to do that. There is always the initial optimization investigation to understand how your assays work. You decide where those thresholds should be, and then you can actually import it into or incorporate it into the software when you set up your plates so that those thresholds are automatic. Yeah, I can't show it to you. I mean, I could pull up the software, but like it's one of those things where you set up a gene, you're like, okay, I don't want you to decide auto sample individually, auto group, which means everybody within that gene. I want to do a threshold set at this. And so it works that way. All right, yes. Okay. Okay. Yes, because when you do the digestion, you have to hope that it's actually breaking up between. You do. Yep. You could, but then you would be competing for the targets. So you might contract the efficiency of the two. That would be an interesting theory, though. Like I'm afraid you would be competing for the same targets in the well. And so the question is what would that really do in terms of the results? It's certainly an interesting theory that would be fun to test. I have not heard anyone that has tried that before. No. Mm-hmm. Mm-hmm. That's true. But by the time you're developing a molecular integrity, you're looking to screen lots of samples versus NGS, which is like $1,000 for one. I think there's the difference there is that if you're looking for just confirmation as a whole what's there, yes, NGS is still your best shot. If you're looking at a long-term project where you're trying to gauge what that kind of looks like, that's when you can utilize digital PCR. It's funny because a lot of people I work with are very much about like, oh, it's only got room for 16 samples. Then I talk to you about how do you do qPCR? Are you running duplicates or triplicates? So if you're running triplicates, that just became one well. So now we've eliminated that. How many targets are you looking for with that same sample? Are you running them multiplex already? Or are you running them individually? So now let's take an example. I'm looking for 3 different targets with my sample. I run triplicates and everything is either FAM or it's Zyra green. Well, that's fabulous. Now I've taken 9 wells and I've moved it to 1. And so it's just a mental concept of the difference of how qPCR is moved to digital PCR. There are ways to make things more effective by utilizing digital PCR. But again, I don't recommend it for just replacing qPCR. Please continue to do qPCR. But I think there is a place where sometimes you've just reached your limits of qPCR detection and you need to think outside the box of how to get the answers you're looking for. And that's something that digital PCR can often do for you. Did I run over already? Are we already set? What time did you start? Okay. Ah, all right, good. Okay. Mm-hmm. I know exactly what you're talking about. Yeah. So I don't have easy access to the slide that I would love to show you at the moment, but we literally ran 12 plates and we checked for consistency across the plate. The coefficient of variation was 1.7% across the 12 plates for the same sample. So the answer is the consistency in our plates, because there's nothing chemical going on. There's nothing unusual. It's very, very consistent across an instrument so that you can rest assured. You can also put them in the study function and take a look at them for variability, but it's really low. Right? Yeah. It is. Yes. So once again, we're going to come back to the fact that digital PCR and qPCR theoretically should give you the same answer. The challenge is we all know that qPCR sometimes reaches limitations when you get down to like, 10 copies or less. There's, what was that? Yes, very much so. So it gives you more sensitivity. The problem is that when digital PCR first came out, everybody was like, wave of the future, qPCR is gone. Yeah, no. First of all, there's always the issue with throughput, right? But not only throughput, there's a cost associated, especially with the original systems back then. It was way more expensive to run your samples on digital than it was to just do qPCR. That's been a bit of a shift at this point. It's less expensive now. But the bigger challenge is that for most systems, rain dance excluded, for most systems, you are dealing with a limitation of partitions. So qPCR is fabulous if you have no idea what you're putting in there, or you have targets that are like on opposite ends of the spectrum, right? We need that range of detection so that we can have GAP, DH, and beta actin, and then we can look for other targets of interest that are low. You just aren't getting the precision you need here. And so my biggest thing, since I've represented qPCR for 14 plus years, 17, I don't even know what the number is anymore. Because I've been doing it for such a long time, I've learned that we've done a lot of things to make sense of qPCR and really push the limits. We moved to probe-based when we're dealing with low expressions, but it's still not perfect. Digital PCR can help you get there if you use it as a complementary tool to to qPCR. At least that's my personal philosophy. The rain dance, I am going to say they were geniuses when they developed that. 150,000 partitions was stunning, especially in the concept of looking for an extremely rare mutation event. They had sensitivity unparalleled to anyone else. It was a crushing blow when they sold to Bio-Rad, and Bio-Rad shut down the system. It was their gravest mistake that they've ever made, because we take trade-ins if you decide you want to try something different. But that was a crushing blow, and it was heartbreaking to see Rain Dance leave, because they utilized the TACMan. They used the probe-based system. Their sensitivity, I won't say, like you said, not necessarily more sensitive. The trick is digital PCR helps you get to where sometimes you can't get with qPCR. Your overall, I mean, depending on your expression levels, I mean, qPCR is just fine. All right, so you all have projects, right? You all want to try something? No, okay. All right, thank you everyone for joining me. Please stick around, because actually we have the pleasure of introducing one of your own who came to the seminar last year and started using the machine and has data of her own to share.